Hello friends, welcome back to my channel BioFreak. Let's talk about DNA polymerases. DNA polymerases catalyze the formation of nucleotide polymers, that is polynucleotides, by joining deoxynucleoside triphosphate via phosphodiester bonds. So basically these are the nitrogen bases, and the nitrogen bases are on top of the sugar phosphate backbone, and the phosphate is basically linked by pre-commodified phosphodiester linkage. DNA polymerases can be of two types, that is template-dependent DNA polymerase and template-independent DNA polymerase. Template-dependent DNA polymerase requires a template. For example, DNA-dependent DNA polymerase will require a DNA template based on which, the DNA will be polymerized or it will be synthesized. And RNA-dependent DNA polymerase, which can be the reverse transcriptase, it requires an RNA template, and based on that, the complementary DNA will be synthesized. Template-independent DNA polymerases do not require any template. Example can be terminal deoxynucleotide transferase. DNA polymerase 1, also known as Conberg enzyme, is monomeric enzyme with low processivity and low polymerization rate. It has these three abilities, that is primer removal ability, DNA repair, and gap filling. So it's because of these three activities on three different domains of 928 amino acid long residue chain of DNA polymerase 1. So the first one is 5-3- isonuclease activity. This is also involved in primer removal. 3-5- isonuclease activity is basically the proofreading activity, and 5-3- polymerase activity synthesizes the DNA. When this particular chain is treated with subtilisin or trypsin, it cleaves it into two fragments, a smaller fragment and a larger fragment, also known as clinofragment. So basically the clinofragment has proofreading activity and polymerase activity, and it does not have any primer removal activity. The next one is DNA polymerase 2. DNA polymerase 2 is monomeric enzyme. It has 5-3- polymerase activity, 3-5- isonuclease or proofreading activity. It has low processivity, low polymerization rate. It also acts as an alternative DNA repair polymerase, and it can replicate the DNA if template itself is damaged. The third one is DNA polymerase 3, the most important one that is involved in the replication. It is, if you look at the structure of this, it is asymmetric in nature, and it has this alpha subunit, epsilon subunit, and theta subunit. This together, it has this core catalytic activity of this particular enzyme. So alpha subunit has polymerase activity. It will, that 5-3- polymerase activity, while epsilon subunit is involved in proofreading activity, that is 3-5- isonuclease activity, while theta subunit, is involved, in helping the epsilon subunit. It stimulates the epsilon subunit's proofreading ability. All these subunits are coded by, different genes as mentioned there. Then we have two beta units which act as sliding DNA clamps. So through this, the DNA, while replication passes, because as I said, DNA polymerase 3 is very important in replication. So one strand, that is the leading strand, will pass through this particular clamp, while the lagging strand requires the clamp loaders. So the clamp loaders, that is the gamma and the other assembly, is actually involved in loading the lagging strand again and again, because we know that there are Okazaki fragments, and then they need to be loaded on this particular clamp, sliding clamp, as the DNA moves further. And the tau unit here at the base helps in dimerization of the core enzymes. So it pulls both the units of the core enzymes together, an asymmetric form, and but the asymmetry is because of the gamma complex here. This is also known as gamma complex. So this is the table, okay, of whatever we just now spoke about and more. So that's all about DNA polymerases.